Before I start, I'd like to thank the organizers for inviting me to this exciting symposium. Today, my talk title is Personalized Cell Therapy for Parkinson's Disease, Hope or Reality. My laboratory has been focused on the study of the dopaminergic system and Parkinson's disease. And as you know, dopamine is synthesized from L-tyrosine by two steps. The first step is tyrosine hydroxylase, which converts L-tyrosine to L-dopa. And second enzyme is L-dopa decarboxylase or aromatic L-amino acid decarboxylase, which converts L-dopa to dopamine. In fact, the dopamine, molecular dopamine, was first identified by Professor Arvid Carlsson in Sweden to be a neurotransmitter in early 1950s, and for which work he got the Nobel Prize in the year 2000. And when I recently checked in the PubMed, amazingly, there are more than 170,000 papers in the PubMed which describe the study about the dopamine. And dopaminergic neurons reside in the substantia nigra and project to the striatum, and this nigrostriatal pathway controls our voluntary movement. Currently, there are about 1.5 million Parkinson's disease patients in the United States only, and worldwide there is 6 to 10 million patients. In the United States, the cost for Parkinson's disease is more than $14 billion, and it is expected to more than double by the year 2040. Age-dependent and selective degeneration of dopamine neurons in the substantia nigra is the pathological hallmark of Parkinson's disease. So the major treatment for Parkinson's disease is a pharmacological replacement by L-dopa or dopamine agonists, but with frequent side effects such as dyskinesia. So there is currently no treatment that can slow down or stop the disease progression. A hallmark pathological feature of Parkinson's disease is age-dependent and selective degeneration of A9 dopamine neuron in the substantia nigra, which makes the Parkinson's disease as one of the most promising target disease for cell therapy. Since 1980s, numerous scientists have tried the transplantation of different types of dopamine-producing cells in diverse animal models, and among these cell types, transplantation of midbrain dopaminergic cells from aborted tissue, aborted fetuses, provided the most promising outcome. Indeed, in early 1990s, many groups showed a very exciting and impressive improvement in PD patients in the open label studies, including the Professor Ola Lindberg in the Lund University. And these studies were published in these top-tier journals. And encouraged by these results, sometimes which show that more than 10 years or up to 20 years of improvement, then there are two double-blind controlled studies in the United States. Unfortunately, however, these studies concluded that the improvement was statistically insignificant and do not recommend the fetal cell transplantation. Furthermore, in the significant number of patients, there was a graft-induced dyskinesia, which eventually put hold about this fetal cell transplantation. In conclusion, these fetal cell transplantation studies demonstrated the proof of principle for Parkinson's disease cell therapy. However, fetal cell transplantation has a significant ethical, practical, and medical limitations to become a general therapy. So the important question is, what could be the ideal cell source for Parkinson's disease? And what could be the ideal cell source for personalized cell therapy? As I mentioned, many people tried the ventral mesencephalic tissue from aborted tissue, and other groups tried the adult stem cell, and in particularly, embryonic stem cell-derived dopamine cells are very extensively studied. Recently, there is a big interest in using direct converted dopamine cells. And also some people study nuclear transfer embryonic stem cell. And my lab has been very interested in using the induced pluripotent stem cell. As you all know, in 2006, Professor Yamanaka published a groundbreaking work by showing that fully differentiated cells, such as a skin cell, can be de-differentiated and reprogrammed all the way back to embryonic stem cell-like cell, which is a so-called iPS cell, which ignited the explosive interest both from scientific and the general public, because this iPS technology can allow patient-specific personalized or autologous cell therapy. Indeed, this iPS technology opened up an unprecedented potential to study and treat human diseases, because it can derive all different types of cells in the body, and they can be used for not only for cell replacement therapy, but other studies, developmental studies, disease mechanism studies, and transplantation studies, as well as drug screening platform. However, this iPS technology is still in the very early stage and not fully translated to the autologous cell therapy. So we identified key issues to address to realize personalized cell therapy for Parkinson's disease. First issue is a safety issue, obviously, and the second issue is a reprogramming technology, and number three is how to optimize in vitro differentiation for scalable production of transplantable cells. And number four is to address in vivo safety and efficacy of the cells in using appropriate animal models. And number five is to optimize neurosurgical procedure. And the last issue is FDA approval and other regulatory and clinical issues. So I will briefly overview each step for our cell therapy application. The first one is obviously safety issue, and there are two aspects in this issue. The number one is how to confirm the genomic integrity of the cells, including normal karyotype, and there should be no host chromosome disruption, and there should be no residual vector DNA, and there should be no harmful mutations, including cancer-causing mutations. And second issue is how can we ensure that there is no remaining undifferentiated cells. After we optimize our reprogramming technology, we generated 4 iPS lines and analyzed them by using whole exome sequencing and whole genome sequencing. And when we compare our data with other human iPS data set and also human embryonic stem cell data set, we found that the genetic burden is much less. And in particular, 2 lines of our iPS clones did not include any non-synonymous somatic mutations associated with the cancer genes. So based on that, we identified the C4 iPS line as one of the most promising clinical grade iPS lines.